Hello, everybody. So we are going to talk about proteins, which is the last biomolecule that we're going to be talking about from chapter 2 here. And so let's jump right into that. By the end of this module, you should be able to describe the building blocks of proteins, describe peptide bonds, explain the structural and functional characteristics of proteins, describe the 4 levels of protein structures, and state what chaperone proteins do, and uh, uh, and why that is important. All right. So proteins are the workhorses of the cell. They do all the work. So our genes that are found in our DNA, which is one of our nucleic acids, they encode for the proteins. And when we think about the processes that go on in the cell, there are proteins involved in all of them at some level. Proteins are polymers, which are made up of individual amino acids. So the amino acids are your monomers. There are 22 of these that are genetically encoded. This image shows 20 of them. These are our standard amino acids. There are 2 non-standard amino acids, selenocysteine and pyrrolysine. It's a mouthful. Now, each amino acid consists of your amine group, which is the nitrogen containing group. This is the inside of your amino acid. And then we've got a carboxyl group, which is the C side of our amino acid, right? And then the thing that makes your 22 amino acids different is this R group. This is the functional group. So the R groups can be broken into a couple of different types. We have our, up here, we can see our electrically charged groups. We have our polar groups. So these are like our nonpolars and our polars. We've got our acidic down here. We've got some special cases over here. Now, these different side groups are what are going to determine the overall structure of the protein. And so to look at them a little bit more closely, here's our non-polar amino acids. Now, for purposes of this class, I don't necessarily need you to memorize the structures of all of these, although if you were to take biochemistry, you most likely will need to know those structures. What I do want you to be aware of is how to differentiate them in terms of we've got glycine, that's GLY. So you'd be able to kind of have some passing familiarity with these different amino acids. And here we've got our polar ones up here. We've got our acidic groups here, our basic here. And then here are our non-standards, the 2 that are not in the big 20. Now, amino acids, when they bind together, they do so in what is called a peptide bond. The peptide bond is when the amino group of one of these binds to the carboxyl group of the other. And so this is the N terminus binding to the C terminus. This is going to form a short chain, which we would call a peptide, and that bond between the 2 amino acids is our peptide bond. If we draw this chain out longer, we would call that a polypeptide. And that's getting to explain our primary structure of a protein, which is that amino acid sequence. Now, once a protein sequence or a protein is made and you've got all the sequence of amino acids, they are going to fold into these different structures. And so we're going to talk a little bit about the differences between the levels of protein structure. We've got a primary, secondary, tertiary, and quaternary structure. And so when we start off, our primary structure is simply that line of amino acids, right? So what is the amino acid sequence? Here, you've got amino acid 1, 2, 3, 4, 5, 6, so forth and so on. And so this is going to be your primary structure. And so you might have a methionine and a valine and an isoleucine or whatever. And this is all going to be determined by those R groups. Now, when we talk about primary structure, this is really the foundation of all the higher order structure. If there's any issues in the amino acid sequence, that can lead to bigger problems down the line. So if you get mutations in your gene sequence leading to a different amino acid to be inserted, then that can cause major problems for the overall protein. Now, our secondary structure is going to be composed of two different primary types. We have our alpha helices, which you get these little spiral structures here that are bound together by these hydrogen bonds, or you get these beta sheets or the beta pleated sheets, which have these little folds that come together because of hydrogen bonds. Next, we have tertiary structure. This is when we start to get the 3D structure of our protein. This is where you're going to start having interactions between your amino acids. So you get ionic and hydrogen bonding, van der Waals interactions. And then for stuff like cysteine, you might build these disulfide bridges like we have here showing the sulfides on the cysteines binding together. And this starts to like fold your protein into a more complex, higher order tertiary structure. Now, lastly, we've got our quaternary structure. Quaternary is when you have two or more polypeptide chains that come together. So you've got two polypeptides that have folded into the tertiary structure, and then they come together to form a protein that is in a quaternary structure. Now, there are all sorts of things that can affect protein folding, including pH and temperature and the different interactions. And some of these proteins are so like their structure is such a high order that they require a chaperone protein, which helps actually mediate that folding so the protein gets the proper shape. So these proteins have multiple functions in the cell. They can be scaffolding for cell. They can be scaffolding within the cell wall or in the cell membrane. There are lots of enzymes that are going to facilitate different chemical reactions, such as breaking or building, molecules. There are cellular transporters that move things throughout the cell. There are proteins that recognize different cell signals or different types of cells. There are proteins that are used for movement, such as flagellin and pillin, which make these external structures. And there are proteins used for communicating back and forth between microbes. So at this point, you should be able to describe the building blocks of proteins, describe peptide bonds, explain the structural and functional characteristics of proteins, and describe the 4 levels of protein structure, and state why chaperone proteins are important and what they do. All right, check out the objective check to see how you're doing on these objectives, and I'll see you soon.